All right, welcome to another week of Soul Seminar Online. Today I'm introducing Julia Dupin. Julia is currently a postdoc at the Department of Ecology and Biological Diversity at the University of Paul Sabatier in Toulouse. She has a project working with Guillaume Baignard on the biogeography and fruit type evolution of the Solanaceae family. She got her PhD at the University of Colorado Boulder, working on phylogenetics, historical biogeography, and the evolution of environmental niche and fruits in the tribe Datureae. So with that, you can share your screen. Julia, go ahead and get started.
Okay. Oops. Oh no. Oh, here we go. All right. Well, thank you, Chelsea, for the intro and for the invite. I'm excited to share this presentation with you guys. I think this is the dream audience for this talk. Okay. So what as Chelsea mentioned, I'm currently at a postdoc at the Université Paul Sabatier in France. But this work that I'm presenting, is a big part of my PhD that I did at CU Boulder. All right, so what I'll present today is my results on the analysis of the historical biogeography and the environmental niche evolution in Datureae, which is a tribe in Solanaceae. All right. So for my work, for this work and for my work in general, I'm mainly interested in trying to understand why plants are found where they're found. And when you look at the in a global scale at clade distribution, different clade distributions, you basically get all sorts of things. But even within all this diversity, we're still able to find some common patterns in geographical distributions. So I have here some examples. So we have plenty of examples of clades that are have a pantropical distribution. So they're mostly found, they're natively found in the tropics. Many others have a mostly northern distribution, and also many examples of clades that have managed to disperse and are native to all continents. So when you start thinking about these distributions and trying to understand them, you basically find yourself trying to identify what are the factors shaping the geographical distribution of these clades. So I'm going to present you here with some of the major factors according to the literature, and for that I'm going to use my toy example here on the side, which this at the bottom here represents a big big land, continental land, and a couple of islands. So the first factor that we should try to keep in mind when thinking about clade distribution is it's the geographical starting point. So depending on where a clade started diversifying and how easy or how close other areas are, that can make a big difference for the distribution of the clade in the future. Related to this first factor, we have the ancestral ecological niche. So how did this area, the starting area, what was the type of environment or the other species that were living in that same area when the species or clay started to diversify. A third factor would be exposure to new niches. And here the idea is that given the starting point, once this one clade starts to disperse to other areas, did it manage to move around far enough to eventually encounter environmental conditions that are contrasting to the ancestral ones? And then what happened after, once this exposure happened? A fourth factor would be to think about any limitations to dispersal. And these limitations, they can be they come in two flavors. They can be abiotic, so just as I mentioned, contrasting environmental conditions. So say changes, differences in the amount of water that is available throughout the year, or you can have a limitation to dispersal that is biotic. So say the clade we're studying is studying in a given environment, and I managed to now disperse to a contrasting one, a different place. But now in this new place, it's an important pollinator is missing, or there's a species that uses the same resource, and now we have competitions. So those things can become with time. They can determine if the species is able to colonize and diversify in this new area or not. And lastly, another main factor to consider is simply the amount of time. So for the clade that you're interested in, once you the simple idea is that the more time it has to diversify, the higher chances of it dispersing and successfully colonizing new areas, and then changing and expanding its geographical distribution. Besides 5 big factors, I think there's 2 other things that are also permeating all of these factors, and should always also be considered for any studies about the distribution of clades. And these would be phylogenetic relationships within the clades that you're interested in, meaning that species that are more closely related will tend to share more of their geographical distribution when you start looking at species, when you compare to species that are now far apart in the tree. So that would be one thing to always keep in mind. And the second thing would be trait lability. If I go back to my example of exposure to new niches, if a clade is now faced with a completely new environment, very contrasting, depending on how labile, for example, some physiological traits are, that can determine if this new environment can be colonized or not. So for my work, for this work and for my work in general, one of my major goals is trying to understand what is the relative importance of all of those factors when you look at the large scale distribution of a clade. So for this work, I focused on Datureae, which is a tribe in Solanaceae. Before I go into Datureae, just a quick reminder on the family. I'm sure that for most of you, this reminder is definitely not needed. But yeah, Solanaceae, so it's a family which has, I think, still an estimated number of 2,800 species, including several economically important ones. Its distribution today, it's in all continents except for Antarctica. That's what my little map here on the top shows. And after our work that got published in 2017, we found out that the family originated in South America, and from this area, it dispersed to different parts of the American continent and then to other continents. And most of the dispersals happened between South America and Central and South America into North America. All right, specifically on the tree. So this is a tribe that has today 20 species described, divided into 3 genera. And this is how the distribution of the genera look today. So out of the 3 genera, the biggest one is Datureae. So Datureae has 12 species, and these species are distributed in the Southwest US, Mexico, and portions of Central America. Brugmansias, so the second genus here, it has 7 described species. It's most of them, 6 of them are along the Andes, are native on the Andean area at different elevations, and we have 1 species that is native from the southeast Brazil in the tropical forest. And lastly, we have the 3rd genus, which is Trompetia, which is a monotypic genus. So it has only 1 species that is found in mid to high elevations areas in Bolivia, in very dry areas of the country. So what some of my questions were for this group regarding its biogeography and niche were what was its geographical starting point, for how long it had been diversifying. Can we say anything about its ancestral niche, and if throughout its diversification, if it presented any shifts in environmental preferences. And specifically, I wanted to test this hypothesis. That was that when species moved from one area to the other, so from area 1 to area 2, this movement would be accompanied by a shift in environmental preferences. And the main idea behind here is that because we were looking at areas that were so far apart, the chances of a long distance dispersal taking the species to now an area that would be very similar to the starting one would be low. And so, in other words, getting to a new area with a new environment would then require a shift in environmental preferences for diversification to continue to happen. All right, before I get into the methods and results, here's some pictures. If you're not familiar with the Datureae, so Daturas have this beautiful white flowers. Sometimes it's white with purple shades. They tend to be upright, and they vary from 5 to 15, 18 centimeters of length. They produce, all the species produce fruits that are classified as capsules. So they usually dry out and open up when the seeds are ready to be dispersed. And they mainly look like this. So they're shrubs that can get up to about a meter of height, and they tend to live in open areas like in the picture here. Brugmansias, they also have some very beautiful flowers, but they, contrasting to the Daturas, they are pendulous, so they point down, and the flowers are much bigger than the Datura ones. So they have flowers that range between the length of the flowers between 18-ish, 20, up to 35 centimeters in length. Also contrasting to the Daturas, they produce fleshy fruits. Berries in this case, although we have no idea what disperses these berries today. And honestly, the fruits look very not interesting and probably toxic for humans, at least. Brugmansias are also described as shrubs, but they are much bigger, much taller than the Daturas. They can get to up to 6, 7 meters of height. And here's one example of a big one. All right, and now Trompetia, contrasting to the Daturas and Brugmansias, it seems like everything shrank down. So the flowers, we don't have a scale here, but this guy is about 3.5 centimeters, so about 10 times smaller than a Brugmansia flower. It's also pendulous. The fruits are also fleshy, also berries. And but round now. The Brugmansia fruits, they tend to be more elongated, and but the Trompetia fruits are round. It's also a shrub, but much smaller. It can get up to 1.5 meters ish of height, and it has these tiny leaves compared to the size of leaves of Brugmansias and Daturas. All right. So what we did. For the historical biogeography, we estimated a likelihood model, which basically tried to given a tree, given the tree for the tribe, it summarized at the nodes what would be the most likely state for each of the ancestors, given the distribution of the species today. So I have my small toy example here, just to explain to you how the results are going to look like. So in my example, I have at the tips the states, so which could be in this case geographic ranges. So we have the blue one and the gray one, and using this type of model, so likelihood model, what we see at the nodes are basically telling us that for this one node, there's a probability of being the blue state of 1 or 100%. But when we look at the second node here, so the second ancestor, so the one for these two species, it's most likely the blue state, but there's also a chance that it was actually the gray state. This is just so you have an idea how the results are going to look like. All right, for the niche evolution, we used, we went back to our phylogeny, but now we use the environmental data. And this data was a combination of bioclimatic layers on temperature and precipitation, but also we included some soil physical properties. By including soil, we were able to expand a little bit on the characterization of the environment. Most studies, they look at the temperature and precipitation, but here we'll be able also to see, for example, if the water retention capacity of a soil, if it could play an important role on the, if we could influence at all the distribution of species or not. For the approach, we did two major approaches. I'm going to talk about the first one here, and then later on the talk I'll introduce the second one. The first one, the idea was just to try to detect any shifts, any changes in environmental preferences when it came to the distribution of the clades. So back to my little examples. So if we have a situation like this, where you have your little clade with the three species and the types of environments where they live today. So one environment where there's a lot of water available, so a wet environment, and one that is more seasonal or drier compared to the first one. We wanted to test if the pattern that we see, that is that one of the lineages living in this contrasting environment, if it happen first, if it's a significantly different type of environment, and second, if this pattern happened by chance, or if it's indeed a significant shift towards a new environment. To do this, all of this, the data that we used, we needed a continuous variable. So what I did was to combine all that information on temperature, precipitation, and soil into one continuous variable using a phylogenetic PCA approach. All right, so I'm gonna jump in straight into my results. So first, I'll present the results on the biogeography. So let me walk you through this figure. So we have here the tree for the tribe with one outgroup, which is Nicandra up here. What we found for the tree is that the Trompetia is sister to the two other genera. So here's the Tura, mainly found in North America, which is the orange color here, with two species having a distribution that is North America plus Central America. Nicandra, Trompetia, and three species of Brugmansia are mainly found in the Andes, so that's the dark blue color. And two species of Brugmansias today, they're found in non-Andean parts of South America. And this can be just low elevations in the northwest portion of South America, or Brazil. So suaviolens is in Brazil, versicolor is in Ecuador. All right, so given this, I'm gonna simplify the tree a little bit. So given the current distribution, the tree, so we estimated the model, and the pies of the nodes, that's what the model gave us. So the first information is that most likely the ancestor of this tribe started diversifying along the Andes, so high elevations or mid elevations in Andean areas. But what happened after that, it's still a bit of a question mark. We don't know if the ancestor for the Turas and Brugmansias, if he was mainly Indian, or if it actually had a distribution that was Indian plus North America. That's what the gray means here. Although the second one sounds more unlikely, but even with that, I just left the question mark here. And this question mark means that we cannot tell specifically where the dispersal to North America happened. So this dispersal from south to north, it could have happened along one of these two branches. All right, so the second information that our model showed was that within Brugmansia, so for Brugmansias, the clades started diversifying in Indian areas, and then you had one major dispersal to non-Indian portions of South America. And here's one example. So the species that diversified in Brazil. And the third major part of the historic biogeography of the Datureae is that some species, in this case two here that we were able to estimate, of the Datureae, so they most likely started diversifying in North America and then expanded their range into Central America, which is this parcel here. All right, when I look into the analysis of niche evolution, our main result was that Brugmansias, they shifted to preferring a more wet type of environment. So I'm back to my little tree here, and besides the shift that we detected in Brugmansia, we also estimated that first, the ancestral state for the tribe is a more dry type of environment, and Trumpetias and all of the Datureae, they live in environments that are drier when compared to the Brugmansia ones. This whole analysis we did in a set of trees, wasn't only on this one. So we tried to manipulate the tree in different ways. So allow it to want to have a set of trees, the topology within the two genera, for example, we changed a little bit. The branch length is also changed. And we also tried to remove taxa from it to see if the main result would be influenced by that, and that was not the case. We always found a shift, and it was always along the stem branch of Brugmansias. So they are this group is living in a significantly different type of environment. So if I go back to my hypothesis that I presented at the very beginning, which was that movements between areas would be coupled with a shift in niche, that is not what we found. The way to look at this hypothesis, the way it would look in the tree, it would basically be an overlap between the dispersal events. So the little white bars here show all the dispersal events that I previously showed you. And if it was true, if the hypothesis was true, the big star I have and at least one of the bars, they will have overlapped on the same branch. Since they do not, that is not this hypothesis is not the correct explanation here. And what we're actually seeing is an example of niche conservatism. So movements between areas, all of the movements we see, even the one that we're not exactly sure in which branch it happened within the same type of environment. So within the blue environment or within the white one in here. And one other thing I would like to point out about the biogeography was that even though the big movement here going from Indian areas in South America to now North America, which it's for the diversification of the Datureae, even though this was a very long movement, what it resulted in the diversification of the genus, it still happened in a similar type of environment. So we went from a dry ancestral area to a new area, but still dry. But even though there was this, the type of environment was conserved, this was followed by very big changes in morphology and physiology. So in the tribe, given that Trumpetias and Brugmansias, they have, for example, pendulous flowers, we have a contrast when we look at the Datureae, that they have the upright ones, the fruits are now dry, the seeds are now specialized to being dispersed by ants. So here's one example of one seed here, and this white portion is it's attached to the seed, and it's very rich in lipids and carbs, so they can attract ants. And so the ants come for that part in the process of taking the seed back to their nest. They help disperse them. And also most of the Daturas, they are annuals when you compare to Trumpetias and the Brugmansias that are perennials. All right, so those are most of the results, but we wanted to do, we had a second approach to the niche evolution analysis. And here what we wanted to see was basically to check if, even considering that one of the clades, in this case Brugmansia, lives in a new or in a significantly different type of environment, does that mean that the niche of all of these different species, does that mean that there's no overlap between them? Or do we see some sort of overlap? Like, could we find a Brugmansia and a Datura and Trompetia living in the same place, for example? So then for that, to test that, we estimated if there was any overlap between the niche of each of the genera. And to do that, we use the definition of niche or niche region that is, it will be defined as being an n-dimensional hypervolume, and the dimensions will be the different variables that we're able to measure. In our case, because for our data, we used a phylogenetic PCA, we're looking at PC components, principal components to represent these variables. So what my results are going to look like is that given a PC, for me, we chose a two-dimensional space because PC1 mainly was explaining most of the distribution. So given a PC1, PC2 volume, which would look like an ellipsis, once you start plotting the different data points for each of the genera, and once you plot it for more than one species or for more than one genus in the same space, if they share any preferences, those two ellipses would overlap. So let me show you the results, and hopefully things will make more sense then. So, all right, so this is what our data points looked like in the PC1, PC2 trait space. So PC1 was the one explaining most of the distribution of the data points, and PC1 was mainly represented by precipitation variables, meaning that most of the data points here, for example, here's all of the Daturas represented in gray triangles. So they're mainly grouped in the area of the plot where higher seasonality is represented. Also, not only higher seasonality, but also less amount of precipitation. So we have precipitation growing from the right to the left. So the Turas, they tended and Trumpetias, they tended to fall on the right side of the graph, and Brugmansias, they mostly spread out a little bit more towards the west, meaning that they were living in areas that had more water available compared to the Turas and Brugmansias. So we compared the different genera, but we have a multitude of data points here, because instead of doing just one data point for each genus, we decided to use each collection point for each of the species, for all of the species as one data point. So we basically transformed the data that we had, so we could give one, we could represent each of the species and each of the individuals for all of the species as one data point here, to be as conservative as possible when representing the niche regions. So I'm going to simplify this figure a little bit, and then we'll look into the overlaps. So if instead of looking at data points, I now look at ellipses, here's how things look like. So for each of the genera, I have one ellipsis here that represents 95 of the data points. And so most, what we would expect as a representation of the niche region, given this space that we're looking at. So we see that Trumpettias, for example, are very much within the niche region of Datureae, and Brugmansias, they have some overlap with these two, but their niche region is much wider, going towards regions with more, where precipitation is more available. For the further explanations, though, I'm not going to talk about Trumpettia, mainly because it only had 5 data points. So any comparisons, any analysis with only 5 data points compared to hundreds of the other two would be very biased. So I'm just gonna talk about Datureae and Brugmansia from here on. So here's a more simplified version of the graph. For Datureae and Brugmansia, so the big ellipses, the open ellipses, they represent the full niche region. But as we saw with the data points, they're grouped in the space. They're not evenly distributed. So I added a second ellipsis for each one of those to represent where within the niche region most of the data points are actually falling. So for our comparisons, what we basically did was that, and I'm going to start from the point of view of the Datureae here, was to consider the full niche region of the Datureae and compare to where within the Brugmansia niche region most of the Brugmansia data points are. And I wanted to see how much overlap that showed. In this case, this overlap is of about 50%, which basically tells me that the probability of finding a Datureae individual in the niche region of Brugmansia is of about 50%. So if you go to most of the places where Datureaes are being found today, there is a 50% chance that a Brugmansia species could also be found there, found living there. If I do it the other way, so if I now look from the Brugmansia point of view, so here's the full niche region of Brugmansia, and here's where most of the Datureae data points are falling within its own niche region. This overlap is much smaller. It's of only 10%. So meaning that if you by chance select an individual of Brugmansia, there's only a 10% chance that it would actually be in an area where Datureaes are living. So when you think about it, it actually makes sense. The fact that Datureaes are being found in so many areas where Brugmansias could be living, and so in so many areas that are have a lot of precipitation, for example. This does make sense because Datureaes have been escaping gardens everywhere in the world, like here in Europe, in different parts of the American continent, in the Asian continent. So they're very good at whenever there's a new environment and there's a little bit more water than they are used to, they thrive very well in those environments. So it is not surprising that there's so much overlap between the places where Datureaes can be found and where Brugmansias are found. But the other way around, when you think about it, Brugmansias are also being cultivated in gardens everywhere, but they're not found, for example, on the roadside, having escaped the garden. So even though their niche region is much wider than a Datureae one, when it comes to environments that are drier or have less water than they need, they don't do as well. All right, so I'm going to bring together all the information I just presented you on niche evolution, but also on the biogeography, and I'll summarize into one figure what we learned about the Datureae and its distribution. So I have a simplified version of my tree with Trumpettia, sister to the other two genera, and just a small map with a reminder where these things are found. So what we found was that at the beginning of the group's diversification, it started diversifying in a more dry type of environment. And here on the side is just a qualitative representation of how dry or wet an environment is. So, and the curves represent the environmental niche preferences of the clade at that moment. So diversification of the tribe started most likely in a drier type of environment. But with time, once time started to pass, so things continued to diversify, and specifically with the ancestors of Brugmansia here, a new type of environment, a new type of niche region is started to be explored also. So we started with the dry one, and now we have something that is a little bit more wet. And if you keep going forward in time, what most likely happen is that the ancestral niche region that was drier continued to get a bit more dry. So the curve is now a bit more to the left. And the new niche region here with the diversification of Brugmansia continue to expand and to move to the right. And finally, what we see today is we have groups within the tribe that have preferences for drier environments, and we have one group that have preferences for more wet and dry environments. But we're still able to see a good overlap between those two regions, so between the preferences of different species. If I bring back my hypothesis, what we found also was that first thing was that the movement between South America into North America, so the diversification of the Datureae, is an example of niche conservatism. So it went from a dry environment now into a new but still dry environment. And when we detected a shift, so an evolution in the environmental preferences, it was detected within the Brugmansia genus, but it happened within the same area. To conclude, I'm to zoom out a little bit. So the results that we found for the Datureae, so the environmental niche conservatism, it's not a surprise. For our analysis of the biogeography of the family, we qualitatively looked at recent range expansions, and most of them also happened within a single type of environment. So the fact that the Datureae did the same, it accords very well with what we see in the family, at least in the last couple of million years. Not only that, what is also a common thing is that, even though plant groups like the Datureae are able to keep their environmental preferences, so have a conserved environmental niche when you move around, this was coupled with a big change big changes in morphology and physiology. With that, I would like to thank you all for listening, and thank you again, Chelsea Rocio, for the invite. I would like to thank my fantastic PhD advisor, Dr. Stacy Smith, and everybody in the Smith Lab that I had the privilege to work with, all my collaborators, all the funding agencies that funded this research. Thank you.
Awesome. Thank you so much, Julia. So now we're open for questions. So if you have a question, you can either make a note that you have a question, or we can write it down. So Frederico actually has a question, so you can unmute yourself, Frederico.
Hi, Julia. It was a great talk. Thank you very much. So I have a couple of questions. The first one is about the niche overlap between the Datura and the Brugmansia, and is if you think that it's also phylogenetically driven in terms of, I think the Brugmansias that are from the Sphaerocarpum clade are more adapted to drier conditions in high mountains. So do you think this overlap is maybe caused by this species especially, or is it in general maybe caused by all Datura, all Brugmansia being able to grow in different environments?
Oh, I think given that what we estimated was that the ancestral niche was more like a dry, I don't think it would be specific for those ones. I think that, you know, it's as part of, as we saw for the Brugmansia niche region, it was like a very large ellipsis compared to the other ones. So I wouldn't think that is specific to for that subclade specific. I think it would be a more like a genus level type
Okay,
of
thanks.
adaptation. Yes.
And the other is about kind of the conservation status. And so forgive me if you mentioned that, but so in your analysis of environments, did you use only South American species, American data, or just also data from the plants growing outside America?
Only in the American continent. Yes.
Okay.
Yes.
Because Datureae also is found everywhere, no?
Yes.
Some species, a few of them, I think.
Yeah.
Okay, and related to that is, so Brugmansia is at least officially stated to be extinct in the wild.
Yes.
I am not sure about it. I think it's kind of difficult question, but what is your take on that, and how do you think that would affect your results? And thank you very much.
Yeah, thank you for the questions. Yeah, I also think it's a hard thing to say that the thing is extinct in the wild. Although it's hard to find them. Sorry, so the question was something like how, if they're mainly cultivated, how that could influence the results, right?
Yeah.
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 I think that even if you consider that they are a lot of the data points are coming from places that were clearly cultivated, or you know they're very near human occupation. I honestly think that the fact that we were able to detect a shift, it shows that, you know, there's something that is, even though there's a lot of cultivation, there's something about the species or the clade that shows that there is some preferences. I think that yeah, as I said earlier, Brugmansias are not, you know, escaping gardens everywhere in the world. So I think even with the cultivation portion of it, you know, bar if they are like in a greenhouse, I think it still shows the signal is still there. Where the fact that we were able to detect a shift so clearly, I think it still shows that there's something about the preferences.
No, I agree, and they continually escape gardens in South America. You don't find them always in gardens. You find them close to humans usually, yeah, but not always in gardens. Usually more like escaped around. So thank you very much.
Yeah, thank you.
All right, Dick has a question, so you can unmute yourself. Dick Olmsted.
Hi, Julia. Thank you. That was really great. I've been a fan of Datureae for a long time. Bob Bai is an old friend of mine. We were undergraduates at the same institution. And Bob, as you know, has done a lot of work on Datureae.
Yes.
I've struggled with the application of biogeobears to biogeographic analyses over the years. It's a great program. It's a really sophisticated program, but I think it can result in confident results that are likely to be in error. And so I want to ask you about that. You left as unresolved the question of when did Datureae disperse from South America to North America? And you had two different branches where you felt that the probability was small enough that you couldn't say with certainty that it occurred at one or the other. But I'm wondering about how those dispersal probabilities are calculated into this. When you look at that tree, you see that the base of that tree, the earliest, the lower node in that tree, is at almost 30 million years ago. long before there was any land connection to South and North America. But the branch leading to Datureae, the North American clade, is down at around 12 to 15 million years ago, when geologists now think there was a land connection and a better migration dispersal route. So one would require a really old dispersal event and a second dispersal event back to South America, also at an older time. And the other would just require just one dispersal event that would be at a time when it would be more likely, given what we understand about the geology today. How can you take into account those sorts of prior probabilities, if you will, about dispersal likelihood when using biogeobears? Because you understand biogeobears better than I do. I have actually never used it myself, although I've been co-author on papers, including with you, that have used it effectively. So, and I would just add that we do have a paper from my lab and press on a different group in which we tried our best to use the character transformation probabilities in biogeobears, and we still came up with the optimal solution that required two dispersal events, intercontinental transoceanic dispersal events, where one was only needed. And so we rejected that and accepted a less optimal solution. And we had to justify that, because the reviewers said, "Why are you doing this?" But I think it was justified.
Okay.
And I would say in Datureae, it looks to me like one event would explain the distribution.
Yeah. So there are a couple of things about biogeobears. One of the main ones is that considering the areas, well, first the number of areas, of units of areas, can influence a lot how the results are going to look like, depending on how many tips you have. So if you have too many units of areas and too many possible combinations, then yeah, there's no way the program is going to be able to converge on a story that makes sense. The second thing is those matrices, when you have to determine like how easy it is to go from area A to area B, they can be also tricky to do. Although I don't know if you on the paper you have right now, if you guys used it, but there is a way to ask the program to try to optimize those matrices a little bit, instead of just taking in the numbers that we're trying to do our best here to say, oh, at 30 million years ago it was X hard to go from South to North America. Just try a little, do some tests, and please help me with this estimate. So the algorithm now can help us a little bit more on that. But if I'm not mistaken, I think you also can, once fossil information is available, you can also put some restrictions to some of the nodes. You can say, yeah, this node definitely cannot have, for example, areas that say it was also in Central America, because Central America wasn't there at this point. So there are some restrictions at that level that you can do also. But it's been a couple of years that I've looked into biogeobears, so I don't know if further parameters or further things that you can tweak would help us with problems like this. Because yeah, in our case, it's clear the chances of an ancestor being in the Andes and in South America at that point in time is extremely low. But I think it was a combination of the number of units I had, how big my tree was, and the restrictions I had on some of the nodes. But yeah, I guess my main answer is yes, it's still the program. It's not able to, I think, it's still not able to give us estimate that would be as good as just biologists just saying, yeah, this one option is not great because we know a little bit more, and it has to be this other explanation. sorry, I don't know if I answered your question
No,
as well as
that's
you want.
I'm happy to hear you talk about it because, you know, like I said, I struggle with the results in cases where I've been involved with using it. And also, you know, in seeing published results that sometimes seem not to make sense, even though a sophisticated algorithm that the user probably doesn't fully understand gives a result, right? That ends up getting published.
But I think I've seen a lot of a fair number of papers where what you described happens is like, you know, we got this result, but we think it's this one, and then you present all the background for it. And then, you know.
Thank you. Good talk. I enjoyed it.
Thanks.
David has a question, so you can unmute yourself.
Hi, Julia. My name is David, and I want to ask you some things. I'm sorry if I missed it, but the first one is what kind of data you use to like make the niche of Brugmansia, herbarium data, or what kind of data?
Yes, it was mainly herbarium, and I also got some of the data from GBIF. But for that one, I tried to be, you know, as conservative as I could be, because, you know, there's so many Brugmansia species that are cultivated in, you know, in so many parts of the world that for the GBIF specifically, I tried to select as well as I could the data points from there. But it was a big chunk, was the herbarium stuff.
Yes, it's very mysterious and difficult to be sure about that, no? Because that you mentioned about cultivation. The other thing is, what do you think about the fruit of Brugmansia, mostly for the section Brugmansia? Because you mentioned that there are fleshy fruits and berries, but I was looking at Datureae ceratojaula, and the fruit looks like a berry, but they are capsules too. So what do you think about the fruits of Brugmansia?
So the Datureae ceratojaula, I think it deserves its own project. It's a whole thing. The fruit is very weird. The plant lives in a very specific type of environment. The flowers, they're usually at a 90 degrees. So I don't know what's happening. So the fruits, they open. They split open at some point, but they do not fully dry before doing that. But as far as I can tell, they are still classified as capsules. But yeah, I also have many questions about the Datureae ceratojaula. I wish I could answer them more precisely, but yeah, this species is crazy.
Yes, it's very interesting, but Brugmansia, it looks like a fleshy and a berry, but sometimes on the fields, I was looking at Brugmansia candida, that is a hybrid, and the walls of the fruit start to ripen, and maybe one time I tried to catch someone for a herbarium sample, and when I touched the tree, and everything was so ripe, and the fruit fall and go down the river, and it's very curious because in Colombia, in the Quindío, Caldas, well, in the central zone, you can see a lot of Brugmansias close to the rivers, but in the classification there are candida. But they are close to the river, and if you go to the river down, you can see more individuals. But I think they say that is dispersed by clones too, but I don't know. That fruit is very interesting too, no? Very mysterious.
So I definitely think that dispersal by water is a secondary type of dispersal for Brugmansias. I have also seen pictures of Brugmansias growing like with branches right on top of a river. So I think as soon as the fruit falls or the seed falls, they're gonna be taken down that river somewhere else. Another thing I know is that the seeds of Brugmansia, they're very corky, so they float very easily. So I don't know if the dispersal mainly happens because the seeds are falling off the fruit, because the fruits I've seen, they completely dry on the plant, and then at some point you see just the structure of the vasculature of the fruit, and then the seeds kind of still in there, and then the fruit and the seeds start falling apart. So I don't know if it's the dispersal is only the little seeds being dispersed, or if parts of the fruits are also dispersed. But I think water definitely plays a big some role, maybe not big, but some role on the dispersal of Brugmansias.
Okay, thank you very much. And
Thank you.
I know you don't talk about the phylogeny, but I would like to ask you what is your opinion about Brugmansia volcanicola? Do you think it's a species or a subspecies of Sanguinea?
So when I did the study, I didn't have a sample for Volcanicola, and for one of our papers, it was actually pointed out that we're missing that species, and but given the work by Hay, he describes it as a species. So when I mentioned at the beginning that Brugmansia has 7 described species, Volcanicola is one of them.
Okay. Thank you very much.
Thank you.
That's all.
Okay. We have another question by Sean. What is your current theory on self-fertilization of the species in Brugmansia, i.e. Brugmansia arbea and sanguinea?
Oh, I do not know. I didn't look at specifically on self-fertilization of the different species. I know that by growing the Datureae in the greenhouse, they did it all the time. They were self-fertilizing, producing fruits all the time. But for the few Brugmansia individuals we had, they never produced any fruit. So what I can say, which is what I've read, is that I don't know, self-fertilization is not a big thing in Brugmansia, but I don't know details about the species or those 2 species in special. I'm sorry.
Awesome. Thanks, Julia. I think that's all the questions we have. If you have any other questions for Julia, I'm sure she's available via email. You can find her on Twitter. Next week, we're going to have Pat Bettinger and Amanda Bose giving us a talk about the dynamics of reproduction barriers in the wild tomato species. So see you next week. Thanks, Julia.
Thanks, Chelsea. Thank you, everybody.